This is Blood and Cancer, the official podcast of MDH Hematology Oncology. I'm the voice of MDH Podcasts, Nick Andrews. Welcome to episode 56. I am Nick Andrews, and I'm joined, as I always am, by Dr. David Henry. The last time Dr. Henry and I saw each other, we were on the train in Washington, D.C., and I said, I think San Francisco is going to win the Super Bowl, and I must apologize for that terrible advice.
What they didn't win?
No, that's what I hear. That's the rumor.
Gosh, no wonder there was a big parade today in Kansas City.
Yeah, Kansas, it's all over the internet, folks. You can see some really interesting stuff if you're interested. But we have a big episode today. Every time we go to one of these big meetings, something really cool happens, and we get together with a lot of thought leaders in hematology oncology. So from the American Society of Hematology meeting in 2019, we have another interview this week. It's with Dr. Brian Hill, and it's with a topic that it's not crazy complex, but it is kind of funky to understand, and that's biosimilars in their clinical setting.
Yeah, I think that's a good way of putting it. I think it's misunderstood and poorly understood. If I think back to my own experience, go back 3-4 years, we never heard of it, and now it's all around. And I think still there's some misunderstanding, which he clarifies. I remember saying to him that it begins with the name. The name is bad because you hear biosimilar and you go, oh, I don't want that. I want the real thing. And as he gets into these drugs are very complicated molecules, folded proteins. If you want to make an aspirin, you go down the aisle at the drugstore and they're all virtually the same because of the chemical structure. These are very complicated molecules that are made in nature's factories, so not in a chem lab or a mass-producing chemistry factory, but some complicated bacteria or fungus, depending on the cell of Chinese hamster ovary cells, make some of the ESAs. And so they come out a little different every time, including we get into rituximab, one of the earliest complicated monoclonal antibodies, has become a biosimilar of itself because it drifts over time. You can't whip these poor cells into making the same thing every time. Yet the effect, our FDA is really very good guidance and requirements that it shows same efficacy with insignificant clinically insignificant impurities that don't cause any problem or toxicity.
It's honestly it's a bit of a revolution, is specifically because I know that there have been promises on discounts. One of my favorite things when in working with hematology and oncology is I like to take a little bit of credit for coming up with metaphors. The metaphor that I would use to describe this to my family or to colleagues or whatever is that you think of it like a grape in a bottle of wine. So you get a Cabernet Sauvignon grape in California, you get a Cabernet grape in France. They're bottled at the same time. It's the same grape scientifically, but these people do this thing, these people do that thing. It's kind of the same, but it's super different. And then if you leave it in the barrel for 10 years, it's completely different, but it's also still the same.
Yeah, good analogy. It's biology and not strict physical chemistry. And they're here. Trastuzumab, trade name of the brand product Herceptin. Now we have 2 biosimilars of those. We have at least 1, 2, 3 filgrastims, whether that's pegfilgrastim or just straight filgrastim. And then the ESAs, the brand name Retacrit comes to mind. So Procrit, Aranesp, Epogen are the brand products. And there is a biosimilar Retacrit, which in my experience, some of our insurances are asking because, of course, it's going to be cheap, right? Well, the promise was like a 30% reduction in price off the brand. And I think you were telling me you've seen information that's about 10%. So it hasn't really lived up to its promise just yet.
Yeah, that's the estimate where we stand right now. And of course, like you go different specialties and there's a bunch of different moving parts to that, but it is certainly nowhere near 30%. But if that's the goal, it doesn't sound unreasonable to me. And what an impact that would make. This is such a complicated but uncomplicated topic. I'm kind of fascinated to see where this goes, but I just can't advocate enough for the fact that calling it a biosimilar, as you mentioned, it's confusing to patients because you kind of walk in the clinic and you say, here, you're going to get the Mr. Pibb because the real one, the Dr. Pepper, either you can't afford it or we can't give it to you based on insurance, but that's not really the case at all.
And, right, and worth pointing out, the FDA is still wrestling with this, but has not allowed it yet. If you go to get your Crestor prescription filled, they may give you generic, and they don't have to tell you. You may notice this and say, well, I want the brand. They say, well, here's your copay now with the brand as opposed to the generic.
Right.
That's possible, but not yet allowed in prescribing these drugs. So if you ask for a brand of one of these drugs which has a biosimilar, you have to get it. And if you ask for the biosimilar, you have to get it. So there's no substitution without permission yet allowed.
So we'll get into that conversation with Dr. Brian Hill. Of course, Dr. Hill is featured in a couple of our ASH episodes. It's a really good conversation. We talk about Herceptin and, of course, acetaminophen and different ways to think about biosimilars in a clinical setting. But coming up after that, clinical correlation with Dr. Ilana Yurkowitz is something really fascinating. When one who doesn't understand the way that oncology works is diagnosed, one of the first questions, and there's literally a movie about this, like, what are my odds of survival? Well, percentages aren't exactly what they seem in oncology and hematology, and Dr. Yurkowitz gets into that this week.
Should be a great discussion because in my experience, that doesn't often come up because, of course, patients and families are afraid of the answer, but somebody will ask and right away in the back of your head, well, there was that trial and it's 30% outcome at 5 years, but this is an individual patient and the 30% at 5 years is an average. So I try and answer, and we'll see what Ilana says, that your outcomes and survival progression-free will be determined by how you respond. You respond well, it's long. You don't respond well, it's not so long, and we need to change therapy.
Absolutely, and it's one of those things where a patient who would want to learn about how this works, the difference between progression-free survival, overall survival, and of course disease-free survival, the percentages doesn't necessarily translate to all these different things, a bunch of moving parts. So really great topic by Dr. Yurkowitz. Again, she is so talented at coming up with small nuggets from daily practice that really do impact the relationship between the patient and the clinician.
And we'll just see how long she can keep identifying new topics. I can't believe it, but
I am suspicious
every
as
week.
well. Yeah, it really is. It's a bit of a streak.
Indeed.
All right, so coming up after the break, we're going to talk to clinical correlation coming up after the break. Coming up next is Dr. Henry's conversation with Dr. Brian Hill from the 2019 annual meeting of the American Society of Hematology.
Welcome to this podcast. You're listening to Blood and Cancer. I'm Dr. David Henry, editor of the online journal mdedge.com/hematology-oncology. And each week we try and bring you an interesting topic for practitioners in blood and cancer. And this week I'm delighted to have with us Brian Hill, Dr. Brian Hill, who's Director of Lymphoid Malignancies at Cleveland Clinic. Brian, thanks for stopping by.
Thanks for having me.
Well, you're welcome. And we want to probe with you a little bit biosimilars. It's been an area that's been of interest in the past year or two, and we'll begin with the unfortunate term biosimilar. To me, many people, when they hear biosimilar, think, well, that's not the same. I want the brand product. Could you say a little bit about what is a biosimilar and how come it's so difficult to make, and the FDA pretty strict guidelines and rules on how to make them?
For sure, thanks. So, when we think about drugs we use clinically, when we prescribe, let's just take an easy example like Tylenol. So acetaminophen has a defined chemical structure, and the molecular weight, the structure, everything is very precise, and you can reproduce that on a chemical basis down to the atom, and those molecules can be approved as what we call generics. And I think most practitioners are very comfortable with interchanging any brand of acetaminophen for any other brand of acetaminophen. It's all chemically identical. What's different with monoclonal antibodies and other biologics is that these are proteins, which are complex macromolecules. It's not just a small molecule, but the molecular weight of these is very large, and they're complicated. They're made in bioreactors. They have modifications that are made in the cells in the bioreactor that creates them.
They're made in nature's factories.
Exactly. Yeah. And so they can have a glycosylation or other post-translational modifications. If you remember all your biochemistry going back, so what I would say about monoclonal antibodies is we're all comfortable using monoclonal antibodies, but if you actually take a batch of, let's just say rituximab, because that's what we're going to talk about, and that's the first monoclonal antibody, as you know. If you could go back to the freezer and pull out the rituximab from 20 years ago and compare it to a batch from 10 years ago and a batch from last year, it's not chemically identical.
Isn't that a great point?
Yeah.
I've heard that before,
Yeah.
and that kind of makes the point.
Right. I think of it a little bit like, your mother's minestrone soup or something, so you know the components, you know the ingredients, you know how it's made, and probably the soup that you had 20 years ago or 10 years ago or last year or today probably tastes about the same if it's the same recipe and the same approach. But certainly if you subjected it to really complex analytics, you would find differences. And so the question with biosimilars, I agree it's not the best term because it suggests that it's not exactly the same. The issue is, does the soup behave the same way? Does it taste the same and have the same effect on our patients?
Yeah.
So with all biosimilars, they have to go through a head-to-head clinical trial comparing it to the originator drug. And before it can even do that, it has to go through a wide array of analytics to satisfy the FDA to even get to the clinical trial. But before biosimilar can be approved, it has to have a head-to-head comparison that shows
With the originator.
with the originator. And so that's been done in the case of rituximab. It's been done for several biosimilars in follicular lymphoma or other B-cell malignancies that shows basically that it performs the same way in terms of response rates and progression-free survival. And
So while
forth.
Europe is way ahead of us, we now are about to have, or have, by the time this is broadcast, this particular podcast, I think 2 rituximabs similar to the brand originator product, and you're comfortable that they performed the same.
Right. Yeah. So I think it's going to be a little bit interesting experience for practitioners, patients, pharmacists, and hospitals, because there may be major price differences between a new biosimilar and the originator. And if your hospital or your pharmacy that you practice with can negotiate a deal to get a major price reduction, this is
This
going
is
to be
where it
better.
might happen. I think the promise of these as they were coming was like a 30% decrease in price, and I haven't quite realized that.
Yeah.
But I think back to your acetaminophen point, I believe the pharmacy can substitute any acetaminophen it wants.
Right.
Is that true with the biosimilars?
Right. So I think that's really going to vary based on pharmacy and therapeutics committees at each hospital. So usually a P&T committee and a head of pharmacy will discuss this. At our institution, we have had those discussions, and already we're finding that an insurance company may approve the biosimilar but not the originator. So in those cases, we actually don't, the patient and the provider don't even have the choice. Mm-hmm. So I think we're going to, in one way or the other, be forced to become comfortable with it.
Well, we're broadcasting or recording now in the end of 2019, and I know in the growth factor area we have Napoleon Alpha, 1 or 2 biosimilars. We have 1 or 2 or 3 filgrastims or 1 pegfilgrastim. So this is, again, very popular in Europe and coming here now. Rituximab, at least 2. Any other monoclonal antibodies you're aware of that might be about to come our way?
My understanding is that Herceptin may be coming as well, and others. Basically, as the patents expire on the older monoclonals, they're going to be coming along. Yeah.
Well, to conclude and restate, biosimilars very carefully vetted by our FDA. The guidelines are very stringent, and they have to get through chemical structure, biologic structure, production process, and then a clinical trial that shows their identical results with insignificant chemical impurities, safety, potency, all the same. And you're comfortable, I certainly am as well, using, in your case with lymphoma expertise, the rituximabs, either brand product or the biosimilars. They're kind of all biosimilars of themselves. Mm-hmm. And so whether it's insurance driven or not, you would use a biosimilar product.
I would, yeah.
Well, I've been talking with Brian Hill from the Cleveland Clinic, Director of Lymphoid Malignancies and Biosimilars, and you're listening to Blood and Cancer podcast coming out each week. And look for us on the online journal mdh.com/hematology-oncology and show notes on that webpage where you can hear bullet points from today's interview. Brian, thanks so much for stopping by.
Thanks for having me.
You're welcome.
And that concludes the conversation portion of our show this week. Don't forget there are show notes available wherever notes are found in your app. Blood and Cancer. We'll be right back with Dr. Ilana Yurkowitz. Welcome back to Blood and Cancer. I am the voice of MDH Podcast's Nick Andrews. It's time now for the human side of hematology and oncology care, Clinical Correlation with Dr. Alana Yurkowitz.
I'm Dr. Alana Yurkowitz, and this is Clinical Correlation, a segment about the human side of hematology and oncology care. Today, I'd like to talk about percentages we sometimes give in oncology, what they mean, what they don't mean, and how they can be interpreted. A while ago, I met a new patient with MGUS, or monoclonal gammopathy of uncertain significance. As our listeners likely know well, this is a condition indicating buildup of an abnormal protein made by plasma cells. It is more common as people age, and it can be a precursor to plasma cell cancers like multiple myeloma. However, the odds of any given individual with MGUS progressing to multiple myeloma, assuming standard risk factors, are low, less than 3% per year. In my experience, this is always reassuring information to patients who are sent to a hematologist-oncologist by a savvy primary care doctor who picked up the abnormal protein. The patient needs to be monitored, but most of my patients with MGUS have not progressed to cancer. Except for one. This patient diligently got his blood work and urine checked by his hematologist every so often, and was extremely surprised and of course crushed by the news that his MGUS had progressed. What was more surprising to me was that he accused our percentages of being wrong. Granted, he may have been looking for someone to blame because he was grappling with the reality of bad news, but he thought our percentages had been overly reassuring. The truth is, our percentages weren't wrong. I explained that they're based on populations. In a given population of 100 people with MGUS, for example, 1 or 2 will progress to multiple myeloma that year. But for the individual, the result is binary. You either progress or you don't. You have cancer or you don't have cancer. You as an individual will get one of 2 results, whether the odds are 50-50 or 99-1. All this may sound obvious, but my patient's reaction reminded me that it can sometimes be hard for people to grasp what exactly percentages do and don't mean. Just as in political polling, if a poll says one candidate has a 90% chance of winning and another a 10% chance, and if the latter wins, it doesn't mean the poll was wrong. My patient's reaction was understandably affected by the emotions that went along with progression of disease, but it made me think about the best way to convey what percentages mean. There's some data showing that pictures can help. For example, showing 10 cartoon people and shading in 1 to show 10%. In addition to this, what struck me with my patient was that it's really about preparation. So it may be worth reinforcing that for the individual patient, the result will still be either a yes or no, and regardless of the percentages, to be prepared for either possibility. Thanks for listening, and I hope you'll tune in next time for Clinical Correlation.
And thank you, Dr. Yurkowitz, as always. That's a wrap on episode 56 of Blood and Cancer, the official podcast of MDH Hematology Oncology. Blood and Cancer is hosted by Dr. David Henry, the editor-in-chief of MDH Hematology Oncology. Blood and Cancer is produced by MDH Hematology Oncology editor Mary Ellen Schneider. All of our podcasts are produced by editor-in-chief of MDH, Dr. Ivan Oransky, executive editor Kathy Skarbeck, and multimedia editor Terry Rudd. Clinical Correlation is written, recorded, produced by Dr. Alana Yurkowitz. I'm your audio engineer, audio editor, and the voice of MDH Podcasts, Nick Andrews. You're listening to Blood and Cancer.